Another promising area of RECNAC research involves immunotherapy. RECNAC research developed a method by which patients' white blood cells can be used to produce an autologous cytokine cocktail, and developed a protocol for administering this cocktail as a biological response modifier for cancer patients. RECNAC research gained the ability to grow dendritic cells and train them with tumor antigens obtained from the patient or produced cheaply in the laboratory. These antigen-trained dendritic cells can be infused into patients to boost tumor-specific immune responses. Throughout our RECNAC project until last July, Neil Riordan, my son, was at the forefront of our research. He was active in developing immunotherapy approaches here and for a period in Costa Rica. Now he comes to us as president of Aiden Incorporated, a company to whom we have licensed our discoveries and patents. Neil will now tell us a bit more about immunotherapy.
Just killing the tumor cells is not, that's not the whole deal. And when we look at the history of spontaneous remissions in cancer patients, well, there was no exogenous agent killing the tumor cells. So what was that? And we had a lot of cases of patients who were receiving vitamin C doses that were well below the concentration that we know is required to kill tumor cells. So there has to be another mechanism. So several years ago, we started looking very heavily at the immune system because that's the only way that we have really to get rid of residual tumor cells if they're killed by chemotherapy, and in the cases of spontaneous remission, it pretty much has to be the immune system. And so we came. This is a very complicated, busy picture, but I'd like to go through it real quickly. This is one type of white blood cell, and it can go either way, this way or this way. And what usually happens when there's a cancer cell in the body or a transformed cell, then the immune system usually takes care of it because there are all kinds of episodes that occur. And usually the immune system takes care of it. And this is the way it takes care of it. This type of white blood cell can recognize a surface feature on a transformed cell or a pre-cancerous cell or a cancerous cell, and it gobbles a little bit of it, and it acts as a messenger cell. And, well, here it gobbles a little piece of it, and then it presents that information. I guess this doesn't work. It presents that information to another type of white blood cell, a T cell, which then, under proper conditions, will proliferate, create a bunch of those clones of that type of white blood cell that's now active against that type of tumor cell and comes back and kills it. So that's the normal event schedule. And in cancer, there's something wrong there. If you have clinical cancer, something has happened somewhere in this cascade. Maybe these cells didn't proliferate. Maybe this cell was not able to engulf any part of the tumor cell. So we focus a lot of our strategies on that, and we've done studies with what are called dendritic cells, these messenger cells, and we've grown large numbers of these up in the laboratory and stimulated them to have the surface features to present to T cells. But I'm not going to get into a lot of that. But this is the schematic of what we've learned in our research and other people's research on the immune system. And the reason I brought this up because I wanted to talk about the two products that Aiden now is going to start producing and what they do in the scheme of things here. And Dr. Mung talked about the plant-based angiogenesis inhibitor. And, of course, what that can do is choke off tumor cells, which is not in this scheme. But what happens when you choke off the blood supply to tumor cells is you wind up with a lot more tumor cells with surface information that is recognized by these cells. Okay? And so it does fit into this scheme. And so you can also directly choke off the nutrient supply to the tumor cells. It's a very good anti-tumor strategy. And we do have a product in the next 2 months will be available to cancer patients which has this the plant extract in it that has this potent angiogenesis, the ability to shut down new blood vessel growth. The other product we have is the bacterial cell wall extract, which stimulates this. It actually stimulates the immune system, and what it does one of the things that we know it does is it stimulates the conversion of this type of cell to this type of cell. So anyway, we have those two products at Aden in Tempe, Arizona, and those will be available pretty soon. The other thing we do is we have a research facility there, and we're continuing to study the immunotherapies that were developed here at the center. And as my father alluded to, one of them is the use of products of white blood cells. We harvest white blood cells from patients, and we stimulate them in the laboratory to produce some soluble proteins called cytokines, which have direct anti-tumor effects and have immune-stimulating effects. And then we give those back to the patients. We have Institutional Review Board approval from the Southwest College of Naturopathic Medicine in Tempe, Arizona, for the use of those cytokines. And so we're doing studies with those right now. We continue work with intravenous vitamin C, and we have pending approvals for the use of dendritic cells to study dendritic cells as immune stimulants in patients with cancer. So we'll carry on the work, and also a significant percentage of the proceeds of this come back to the center, to the Biocommunications Research Institute, to fund further research. So that's what we're up to, and